Hello everyone. In this chapter, we will look into the information contained in the human genome. As we cover this chapter, we will focus on 5 learning outcomes. In the first outcome, we will try to understand and explain what genomics is and what it is not. Then we will define key terms that are commonly used in genetics. The third outcome will focus on understanding the different levels of genetics. Then we will dive into some applications of genetics and genomics. And finally, we will try to have an understanding of the global perspective on genomes. So, the first question we would like to ask is the following. What is genetics? Genetics, it's the study of inherited traits such as the color of your eyes, the color of your hair, your height, or even your ability to break down lactose when you drink milk. Genetics is also the study of the traits variations and their transmission from parents to offsprings from generation to generation. Genetics is a very powerful source of information about our identity because it can reveal information about our past and potential diseases we may be prone to. Human genetics touches many aspects of our lives and also our community, including forensics, bioethics, psychology, and even history. Today, human genetics touches even consumer genetics, which is basically when you are able to obtain some DNA from yourself and be able to find out a number of information from analyzing that DNA. For instance, if you swab some cells from your cheeks, you can isolate DNA from these cells, and after isolating DNA from these cells, they will be analyzed and find out a bunch of information about people you didn't know you were related to. You can find where you come from. For instance, you can find that you have 42% of your genome that can be traced back to Italy, that you have about 24% of DNA that comes from Ethiopia and Swedish, and so on and so forth. For those living in America, you can maybe find out that your ancestry are from Native American, information you maybe didn't know. You can find out that you may be prone to depression and that some drugs would be a better fit for you to treat a particular type of disease than others. You may find out that you are at risk of developing a number of cancers, such as breast cancer, colon cancer, prostate cancer, and so on. With that being said, it is very important to understand that genetics should not be confused with genealogy. Genealogy actually considers your relationships without taking into account the traits that may exist between people in this relationship. Now, today we talk more about genetics genealogy that examines how people are related to each other. So we don't just say that they are related. The question is, how are they related? Can we prove that they are related using genetic information? Another aspect that is very important to keep in mind as we address genetics is heredity. Heredity is the transmission of traits and biological information from one generation to another generation, right? From parents to offsprings. Also, it is a fact that certain difficult to define human characteristics might appear to be inherited if they affect several family members. Nevertheless, we see that these features may be occurring because of shared genetic background, but also very much the fact of environmental factors. At this point, let's define some terms that are commonly used in genetics. The first stop we are going to make is defining genes. Genes are sets of instructions that are going to cause the production of proteins. Proteins which actually is the visible part of our genetic inheritance, right? Genes are composed of DNA, also called deoxyribonucleic acid. That deoxyribonucleic acid that is made up of nucleotides, we are going to see that later, is the key to holding the information necessary for the production of proteins. The next term we would like to define is the term genome. Genome refers to the complete set of genetic information in your cells. Then we may ask, well, what are cells? The cells in your body are the basic unit of life. They are typically going to contain two genomes. One that you receive from your mom and one that you receive from dad, right? Now, we may ask, okay, well, why do we call the cells the basic unit of life? Well, the cell is the smallest part in your body that has all the characteristics of life, meaning they can reproduce, they can respond to the environment, they can use energy, they can basically interact with each other, and so on. The next term that we are going to address is going to be exomes. The exome, it's a part of the genome that is going to specifically produce a protein. While genomics is a discipline in biology or in genetics that compares and analyzes the functions of many genes across living systems. And finally, but not least, we are going to define bioethics. Bioethics addresses issues and controversies that arise because of the application of medical technology and the use of genetic information For instance, when we isolate the genetic information from someone and we store that genetic information, what are we going to use that information for? Are we going to use that information to basically profiling people? Or are we going to use that genetic information for in a more respectful way, right? With respecting the limits of what we can do and what we cannot do. So now that we've defined some terms of genetics, let's take a look at the different levels of genetics. Now, the very basic level is going to be the base pair. Now, DNA is a molecule that is made up of sequences of A, G, T, and C. Adenine, guanine, thymine, and cytosine. That sequence of DNA is the very basis of the formation of genes. And the genes are going to be packed in a long DNA molecule. And that DNA molecule in the cell is going to be combined with some proteins here. And these proteins are called histones. These histones allow the DNA to be compacted and form a very compiled structure that we call chromosome. The chromosomes are going to be in many shapes and size in our cells, and these chromosomes are going to be stacked in the nucleus, which is right here, right? And basically these chromosomes in a cell are going to constitute your genome. And that genome, when you consider many individuals in a given geographical area, these individuals are going to come together to form populations. And from populations, we can go on and looking at genetics at a point where we are trying to understand how organisms from one species are connected with organisms from a different species. So basically, we are going to take a look at the evolutionary aspect of how organisms are connected to other organisms. Now, let's take a look at DNA and let's take a look at the structure and the function of that DNA. Now, DNA stands for deoxyribonucleic acid. I want you to remember that. DNA, it's a double-stranded molecule. Why double-stranded molecule? Because we see that here you have one strand that is wrapping around another strand. So this is a double helix. Now this double-stranded molecule is made up of nucleotides. Nucleotides are the building blocks of DNA. And these nucleotides are going to be made up of three parts. One is the phosphate group that we can see right here. Two, it's a 5-carbon sugar. In this very case, the 5-carbon sugar is called deoxyribose. This is the 5-carbon sugar. And finally, we're going to have the nitrogenous base. In this nucleotide, the nitrogenous base, is right here. So you have three parts that make up a nucleotide: a phosphate group, a 5-carbon sugar that is called the oxyribose, and a nitrogenous base. Now, there are 4 types of nitrogenous bases that are found in DNA. We do have adenine, abbreviated A, cytosine, abbreviated C, thymine, abbreviated T, and guanine, abbreviated G. Now, when we consider the double helix of DNA, we notice that A always pairs with T, while C always pairs with G. And you can see that here, where G pairs with C and A pairs with T. So, because of that, again, DNA, it's a double helix molecule, right? A double-stranded molecule. When you know the sequence of one strand of DNA, you can deduct the sequence of another strand of DNA. For instance, here, if we look at this strand here, we have C, T, C, T, C, G, A. Now, remember that A pairs with T and C pairs with G. So for this C right here, the complementary is going to be G. For this T right here, the complementary nitrogenous base is going to be A right here. For this C, the complementary base is going to be G, and so on. Now, there are a number of characteristics that are very typical to DNA. 2 very specific characteristics that I want you to remember. One is that DNA can replicate itself. Because it can replicate itself, you have that during the cell cycle, the genetic information is copied so that the daughter cells are going to inherit half of that replicated DNA so that they can have, at the end of the day, the exact same genetic information in the new daughter cells. For instance, when your skin cells multiply, or when your liver cells multiply, right? These cells are 100% genetically identical because the genome is replicated during the cell cycle. The second thing that is very important in DNA to consider is the fact that the information contained in DNA can be used to manufacture proteins. These proteins are the very components that can be seen when they are expressed, when the genes are expressed. Now, let's take a look at gene expression. And for that gene expression, we are going to take the example of one particular gene of interest to us. It's the gene that encodes a transmembrane protein that we call cystic fibrosis transmembrane conductance regulator, CFTR. The gene that is responsible for the production of that protein is carried by chromosome 7. That chromosome 7 in our cells is going to carry the gene. That gene is going to be read and be converted to protein through a process that we call gene expression. Now, gene expression starts with converting DNA into RNA through a process that we call transcription. And from there, when we have RNA, and in this particular instance, messenger RNA, why do we call it messenger RNA? We call it messenger RNA because that RNA carries the information necessary for the production of the protein. So the messenger RNA, through a process that we call translation, is going to cause a production of sequence of amino acids. That sequence of amino acid is the protein. That protein is going to undergo folding. So it goes from primary structure to tertiary structure. And that tertiary structure, it's nothing but a 3D shape. That 3D shape is essential for the function of that protein. Now, once the protein is produced, it adopts a 3D shape. That protein is going to come and embed in the plasma membrane. So here is the plasma membrane here. It's a phospholipid bilayer, okay? And as the protein embeds in the plasma membrane, it's going to form a channel. That channel is the mean by which the cell is going to control the movement of chloride ions from the inside to outside and from the outside to inside. So this allows to control basically a water-salt balance. Nevertheless, we know that that gene is not always stable in all individuals, in every people. Some people have that gene that is mutated, that is changed. A mutation, it's basically a permanent change that occurs in the sequence of DNA. Whenever that a change occurs, this is going to cause a gene not to exist in multiple variants in a population, right? Your mom, for instance, may have a certain sequence of the gene CFTR, while your dad has a different sequence. That doesn't really mean that the gene is different. That just simply means that there are some slight modifications so that the overall aspect of the gene is the same. But this slight modification causes us to now come with another term when addressing these genes. We use the term alleles. Alleles are variants of the same gene, right? And this variant exists because of mutations that can occur. And these mutations can be punctual mutations where you have a replacement of one nucleotide with another nucleotide. For instance, A can be replaced by C, or G can be replaced by T, and so on. And when that mutation is passed down from parents to offsprings and to down the generation, that mutation becomes permanent in the humankind, right? Now, typically, mutations are going to be passed down whenever these mutations occur in sperm cells or in egg cells. This is when they are able to be passed down to offsprings, because you know that in human reproduction, an egg cell is going to be fertilized by a sperm cell. So your skin cells have nothing to do in your reproduction. Your muscle cells have nothing to do in your reproduction. So even if your muscle cell is modified, in as much as your sperm cells are fine and your egg cells are fine, there is not going to be any mutation passed down the offsprings. Okay. Another dimension that is very important to remember is that this mutation that occur, the different alleles, right? This mutation can be positive whenever we consider a mutation to be positive. We say that there is a gain of a function. A mutation can have as a consequence a negative consequence, which is that the mutated gene is going to lose its function, or neutral, meaning that there is no change at the end of the day when it comes to the function of the protein. So now, we saw cystic fibrosis protein CFTR. Know that cystic fibrosis, it's a health condition in which the CFTR gene is modified. Now, when the CFTR gene is modified and the modified alleles are expressed in an individual, then this leads to a number of consequences, including the sinuses that can show some inflammation. Sinuses can be infected, or even some polyps can develop in sinuses. The airways can be affected with a large amount of mucus that develops in the airways, causing an obstruction of airways and also causing repeated respiratory infections. Another organ that can be affected is the liver, where the ducts where biles are produced and move through can be affected. Now, if the bile salt cannot move properly across the ducts, this means that the fatty acids, the lipids are going to be a hard time. You're going to have a hard time digesting the lipids. Let's take a look at the pancreas. The pancreas can also be affected whenever we are expressing the mutated form of the CFTR protein. Well, the ducts that are supposed to allow digestive proteins to move through may not allow this digestive proteins to move through. We have proteins that are responsible, enzymes that are responsible to digest carbohydrate, digest proteins, digest the lipids, and nucleic acids. Well, without those digestive enzymes, we are going to have a major issue, right? And also, the pancreas happens to be an organ that controls also the amount of sugar in our blood. The pancreas produces two hormones. One is called insulin, the other one is called glucagon. Both control the amount of sugar in the blood, right? Insulin decreases the amount of sugar in the blood, while glucagon increases the amount of sugar in the blood. Now, if the pancreas is not producing or releasing rather this hormones because of the ducts that are blocked, now we have an issue, right? The intestine can be affected in people with cystic fibrosis. Our reproductive tract can also be affected, and this is particularly important in males because the duct difference where sperm are going to move through to during ejaculation to actually travel and go fertilize the egg. Now, if the duct difference is obstructed, you are not going to be able to release sperm cells. Therefore, the individual with cystic fibrosis may have a limited possibility for fertility when they are male. And finally, but not least, you have a salty sweat that occur in individual with cystic fibrosis. So now that we've looked at some organs that can be affected because of a mutated version of CFTR, let's take a look at what specifically is mutated in CFTR. Well, in the wild type gene of CFTR, in the sequence of DNA, we are going to have C, C, A. These three nucleotides together, when they are converted into RNA, they give GGU. And this GGU actually is going to cause the production of glycine, which is an amino acid. And when we have a wild type copy of CFTR with the presence of glycine, we're going to have a nice channel that is going to be embedded in the plasma membrane, allowing therefore a proper movement of chloride ion in and out of the cell and maintain a normal amount of mucus across the membrane. However, when CFTR gene is mutated, in this particular instance, C is replaced by T. The consequence of that in the messenger RNA is that we're going to have GAU instead of GGU. That change in terms of amino acid sequence is going to cause the replacement of glycine with aspartic acid. Now, aspartic acid has different chemical properties compared to glycine. Because of that, well, rather than forming a channel, the CFTR mutated protein is not going to form a channel, is going to form a protein that is still folded in 3D, but that doesn't have a channel. Therefore, the chloride ions here are going to be blocked. And because the chloride ions are blocked, now we're going to have an unusually high amount of mucus outside of the cell, which is a major issue in cystic fibrosis patients. Now, at this point, we are going to take a look at the chromosome and much more. When we look at our cells and we isolate the chromosomes from the DNA, we notice that looking at that DNA is combined with proteins. The combination of DNA and protein cause chromosomes to exist, which is a compacted form of DNA. Okay? Now, during the cell cycle, the DNA is not always compacted, condensed in a visible manner, right? Most of the time, it's actually unwound. And when that DNA and protein is unwound, we actually don't call that complex structure a chromosome. We call it rather chromatin. Chromatin. That chromatin, it's in a form that allows gene expression, right? Genes can be expressed. But let me close that parenthesis. When we consider the cells in our body, in your body, you really have two major categories of cells. You have what we call somatic cells, or cells of the body, and you have reproductive cells. Okay, reproductive cells. Reproductive cells. These reproductive cells are also called gametes. All right, now, the somatic cells in your body, for example, your skin cells, your liver cells, your bone cells, your white blood cells, they have 46 chromosomes or 23 pairs of chromosomes. 23 that you receive from your mom and 23 that you receive from your dad. So together it's 46. Out of this 23 pairs of chromosomes, you are going to have 22 pairs of chromosomes that we call autosomes. These autosomes are chromosomes that are not associated with sex characteristics. For instance, the development of breast, okay, the development of hair in your pubic area, in your armpits, right? So the 22 pairs of autosomes are not associated with sex characteristics. Then you have one pair of sex chromosomes. These sex chromosomes in humans, X and Y, are going to be responsible for your sexual characteristics, right? A female individual is going to have 2 X chromosomes, while a male in human species is going to have 1 X and 1 Y. Now, when we take a somatic cell, we can isolate the chromosomes and we can look at these chromosomes. But before we dive into that, I want to say that the reproductive cells or gametes, they contain only half the number of chromosomes you will find in somatic cells. Now, these reproductive cells are said to be haploid. They are haploid because they only contain half the number of chromosomes, while the somatic cells are diploid because they have 2 times the 23 chromosomes. With that said, when we analyze the chromosomes in somatic cells, we can actually develop a map that we call karyotype that is going to display the chromosomes by pair, right? So here we have a karyotype, right, of a female. Why? How do we know this is the karyotype of a female? Because we do have an X and an X here. So this individual has to be female. These are the sex chromosomes, right? X and X. So we have pair number 1, one that you receive from your mom and one you receive from your dad, right? That's pair number 1, pair number 2, pair number 3, number 4, and so on and so forth. When you look at this karyotype, notice that the chromosomes are positioned in such a way that they are put from the longest to the shortest set of chromosomes. So with a karyotype, you can tell the chromosome apart. That's one. Two, with the karyotype, you can tell if an individual has extra chromosomes. That is the reason why when you want to find out if someone or an embryo will develop trisomy, you can isolate embryonic cells, do a karyotype, and see that, for instance, you have an extra chromosome in position 21 here, and that would be trisomy 21 or Down syndrome, right? So with a karyotype, you can tell if someone has an extra chromosome or not. You can tell if a set of chromosome is unusually long or unusually short. However, with a karyotype, you can never tell if there is a punctual mutation, if there is the replacement of one nucleotide, for instance A, with a nucleotide, for instance C, right? You cannot say that. Now, let's try to understand traits. First of all, what do we call traits? Traits are features that can be measured, that can be observed, right? Now, obviously, they start with genes, and these genes are expressed and produce proteins. From these proteins, we are going to have what we call a phenotype. But that's another story. We are going to address that. Whenever a trait is controlled by one single gene, we say that this trait is a Mendelian trait. For instance, when someone has a pointy line on the head that we call widow's peak, right? Widow's peak is controlled by one single gene. Freckles is controlled by one single gene. Long fingers, short fingers is controlled by one single gene. So all these examples are examples of Mendelian traits. However, most characteristics in our body are going to be complex traits. But why do we call them complex traits? We call them complex traits because they are either controlled by more than one gene or controlled by more than one gene and environmental factors. For sure, most of the features that we see in our body are controlled by either multiple genes or combinations of multiple genes and environmental factors. The more factors that contribute to a trait or illness, the more difficult it is to predict the risk of occurrence in a particular family member. Makes sense, right? We can have the same family origin and grow up in completely different environments, right? Because we were isolated at birth, right? These are conditions that can affect our what we are going to exhibit. The type of food we eat, right, is going to affect how we respond to how we express our features. One example of a Mendelian trait, polydactyly, right, which is having an extra finger in the hand or extra toes. It's a Mendelian trait, while hair color is a multifactorial trait because you have at least 3 genes that are responsible for the color of your hair. But even then, you have the environment. Example, the amount of sun you are exposed to throughout a year, is going to affect the overall color of your hair. Now, we looked at cells, we looked at the genetic information inside the cells. Now, let's take a look at a more complex structure. So, we are going to go from cells to tissues to organs. A human body is very complex and has about 30 trillions of cells. These cells occur in different tapes. Not tapes, but in different shapes, and also play different roles, right? You have, for instance, your muscle cells, you have your nerve cells, you have your white blood cells. All the cells in your body, they look different, they do different job, yet they contain the exact same genetic information. Your white blood cells and your nerve cells contain exactly the same genetic information, 23 pairs of chromosomes. The question is, well, if they have the exact same genetic information, why are they different in structure and in function? The answer is differentiation or specialization. Well, let's step back a minute. What is differentiation? Well, what happens is that in the muscle cell, compared to a nerve cell, you have a number of genes that are expressed in muscle cells that are not expressed in nerve cells. Likewise, you have some genes that are expressed in nerve cells that are not expressed in muscle cells. As a consequence of that, you are going to have sets of proteins that are more abundant or present in muscle cells that are not present in nerve cells, and vice versa. Because of that, the shape of the function of these two kinds of cells are going to be completely different. So, differentiation exists because of what we call differential gene expression. Differential gene expression, meaning that the sets of genes that are expressed in a muscle cell is not the same as the sets of genes that are expressed in nerve cells, and vice versa. Okay? So, now, muscle cells, nerve cells, skin cells, liver cells, white blood cells, red blood cells, they are all different because of that differential gene expression. Okay? And these cells are differentiated. However, before the cells differentiate, they start as stem cells. Stem cells are basically blank cells. Cells that can become any other type of cell whenever they receive a signal to differentiate into that very type of cell. Okay, so the stem cells are going to multiply and renew constantly so that you can have blank cells from where differentiated cells are going to develop out of. For instance, if you consider the cells in your red bone marrow at the extremity of long bones, there you are going to have stem cells. And these stem cells, when they receive the signal from a hormone called EPO, which stands for erythropoietin Poietin. Erythropoietin is a hormone that is released by the kidney. When erythropoietin is released, that hormone travels in the blood, goes in the red bone marrow at the extremity of long bones, and cause stem cells in the red bone marrow to turn into red blood cells. Okay, so that is EPO. It's a signal for the differentiation of stem cells in the red bone marrow to become red blood cells. Now, differentiated cells that are similar are going to come together to form tissues so that they can work toward the same function. And more than one tissue. Now, what are the different tissues? Let me tell you about the different tissues. We're going to have muscle tissues. We're going to have nervous tissue. We are going to have connective tissues. And finally, we're going to have epithelial tissues. These are the major categories of tissues in the human body. Now, more than one tissues are going to come together to form organs. And these organs are going to work toward the very same function. And organs are going to come together to form organ systems. For instance, the circulatory system, the respiratory system, the digestive system, and the muscular system. So every single system works as sets of organs toward a specific goal. At this point, we are going to take a look at individuals, families, and eventually down, we're going to talk about population. Now, one thing that is very important that just popped in my mind right now, what I didn't tell you is that among the somatic cells, the cells of your body that have 46 chromosomes, right? There is only one category of cell that doesn't have 46 chromosomes. Actually, they have almost no DNA. These are red blood cells. I want you to remember that red blood cells do not have DNA. Because they do not have DNA, red blood cells are not able to reproduce. Let me close that parenthesis. So now, at this point, when you consider an individual, that individual is going to have two sets of chromosomes, right? One they receive from mom and one they receive from dad. Now, these sets of chromosomes are going to carry a copy of a given gene. For instance, the CFTR gene. For the CFTR gene, you receive a copy from mom and a copy from dad. And remember, that gene can occur in alternate forms, right? That we call alleles. So you're going to have one allele you receive from mom and one allele you receive from dad. Together, these two alleles are going to constitute your genetic makeup or genotype. That genotype is going to be expressed into protein and cause some features to develop, right? Some features that are observable, right? They can be quantifiable. They can be measured, right? This is what we call phenotype. So the phenotypes are the visible aspect of the genotype, right? You explain the phenotype by the genotype of an individual, right? Now, because alleles occur in variant forms, we notice that some alleles are dominant while others are recessive. Alleles that are dominant are always expressed when they are in a genotype. You are going to have some individuals that are going to have two dominant alleles. Some individuals are going to have, well, when they have two dominant alleles, they receive one from mom and one from dad. Some individuals are going to have one dominant allele and one recessive allele. In which case the dominant allele is going to express, you never see the recessive allele express. The only time a recessive allele is going to express, it's when an individual has a genotype that is made up of two recessive alleles. Two recessive alleles. Okay, well, I need to, oops. Oh, come on. Are you kidding me? So next we have the pedigree. The pedigree is a chart that shows how members in a family are related and how they inherit some genetic information from parents. Okay? So in a pedigree, we are going to see that offsprings are going to have half the amount of genetics of the pedigree is going to show how members of a family are connected, and it's going to also show how individuals are going to obtain a particular trait that we are considering, right? For instance, when you consider this pedigree here you are, you have half the amount of genetic information that your parent has, right? You are 50% of your mom and 50% of your dad, right? But now, when we go to your grandparents, the genetic information that is found in you only represent 25% of your grandparents. So you and your grandparents, you are 25% similar, if you will, in terms of the genetic information that is present in your cells, right? With your great-grandparents, it's going to be 12.5% of similarity, right? So the pedigree allows us to tell how much information, genetic information, you are going to share with people in your pedigree line, your pedigree chart. Now let's take a look at a population. So we looked at individuals, how individuals are connected to other individual in your family, right? But then many individuals are going to come together to form a population in a given geographical area. But when we talk about population, really we have to consider from which point of view are we addressing population. There are two aspects when it comes to biology. You have the biological definition of a population. In a biological definition of a population, it's a group of individuals that can have healthy offsprings, and these healthy offsprings can themselves have other offsprings down the line. That's the biological definition of population that does not imply the traits. When it comes to genetics, though, the genetic definition of a population, it's that a population is simply a large collection of alleles for a given gene, right? A large collection of alleles, okay? That population is going to have a very specific frequency of a particular allele in that population. Now, as you compare two populations, say for instance, the population of human that lives in Tuscarawas County and the population of human that lives in Cuyahoga County. When you compare this population and you look at cystic fibrosis, for instance, we are going to ask, okay, well, let's consider the occurrence of cystic fibrosis, the occurrence of the allele of cystic fibrosis in the population from Tuscarawas County versus the population of Cuyahoga County. We could notice that the frequency of cystic fibrosis is different, in which case we are going to say that, well, Tuscarawas County versus Cuyahoga County, it's two complete separate types of populations, right? Because the frequency of the allele cystic fibrosis is different, right? And now, when we consider that population, though, at a genetic level, we are going to call the collection of allele gene pool. That gene pool is what is characteristic of a given population. And now from population, we can compare genomes and show evolutionary relationships. For instance, we can show that populations of Caucasian in America can be traced back to populations in Europe, right? That's some level of evolutionary relationship, right? One can wonder, well, Dr. Jean, why do you say evolutionary relationship when we are still in the same species? Well, believe it or not, there are many changes that occur as you move populations from one area to another area, forming pockets of populations. So the evolution is going to happen differently for people living in America compared to people living in Europe or Africa or Asia or Australia, right? We know that because mutations occur. But let me close that parenthesis and let's be more global, right? Let's be more global. So at that point, when comparing genomes, we can actually literally go across species and show that, for instance, when we look at the genome of human and the genome of chimpanzee, there are a lot of similarities, but there are also some differences. But in terms of similarities, we're going to be like, it looks like they have somehow a relationship, right? So at this point, we are going to dive into the applications of genetics and genomics. Because of all the knowledge we have gathered about genetics and genomics, we now use that information for many purposes, including healthcare choices. For instance, which treatment is going to be given to a particular patient, okay? Genetics is going to impact what we eat, right? We know that the corn in the United States is very much genetically modified, right? That corn is going to be fairly different from the corn that you are going to find in my home country, Gabon, okay? Because simply we are not using the same source of grains to produce corn, right? Genetics can also impact our past and even our future. Today we talk more and more about citizen scientists. Citizen scientists, it's basically when you as a citizen, you become curious about your genetic background. You want to know where you come from. You want to know if you are at risk of developing some cancers. You want to know if a treatment is going to be better suited for you or not. You gather all this information and you decide to interact with scientists and share your data with scientists. And scientists now can use this data and basically enrich the scientific knowledge that we have about human genetics, right? Now, with all these applications that affect not only you, but also your environment and people around you, it is worth asking the following question though. When you hear genetics, what comes to your mind? Do you feel fear? Do you feel a sense of hope? Anger? Are you wondering what is going to happen now? Or tomorrow? Is there a sense of despair when you hear that word genetics? What comes to your mind? It's very important to keep to think about this thing because they are very much part of your life today. Another question that I want you to think about is how is DNA information used today? What is it used for? Is it used for profiling people, to track some people down, to pin some crimes on people that they didn't commit? Is it used to show that some people that were thought to be the criminals are not criminals? How is the information used, right? Even in citizen scientists, right? How is the information used? And then more close to us, not to say that what we said so far is not close to us, but a little more close to us, we had a pandemic that we just went over and we ended up developing a vaccine, and that vaccine played a tremendous role. One can wonder, and actually a lot of the questions I received, because people know I'm a microbiologist, and they ask me, well, Dr. Jean, we are suspicious about this vaccine. How is it possible that this vaccine was developed so fast? Well, let's backtrack a little bit. In the year 2002, 2003, there was an outbreak of coronavirus in Asia, and we also saw some spills in Europe and in America. However, that outbreak never became a pandemic. Another outbreak occurred in the year 2012 in Middle East, of coronavirus I mean. Now, mind that since then, researchers, microbiologists, and very specifically virologists interested in coronavirus had been doing bunch of research. But remember, these outbreaks happened long time ago. That's one. Two, no one cared about coronavirus at that point. And scientists, being stubborn and doing what they do, they keep doing their research and they find ways to deal with the problem. Except that to the eyes of most of the people, nothing is happening, right? Well, guess what? When COVID-19 occurred, scientists who were working on coronavirus had already a tremendous amount of information. They had already tools they had developed. But at the time, no one was interested in that. When coronavirus became a problem in 2019, 2020, well, and then all of a sudden, government starts saying, well, we need to solve this problem. Question, what tools do we have? Well, guess what? Scientists who had been working on this on this topic for a fair amount of time said, well, we have solutions, except that we don't have the money to make that solution available to the most people. And this is how the Pfizer vaccine, the Moderna vaccines were produced. So it's not some kind of conspiracy or people just bypassing procedures for the production of vaccines. Scientists had a lot of information way before the pandemic, but no one cared about that information at that time, and no one wanted to know anything about it, right? But now that we were in a situation of the pandemic, well, people were like, okay, well, we better do something because our economy is falling down. We are all imprisoned in our own houses, right? And we don't see the outside anymore. We need to do something. So once the politician recognized that something needs to happen, then the scientific labs and scientists started receiving a bunch of money, and now they could, upscale everything they were already doing. So let's look a little more about comparing DNA and showing relationships. And again, this is going to be on applications of genetics. So being able to compare DNA is going to show relationships between individuals. DNA profiling, it's a technique that is used today in forensic science to show who was present at the crime scene, who was actually the criminal, who was not the criminal, right? With DNA profiling, it is possible today to identify victims of accidents or natural disaster. Think about, for instance, what happened, the tsunami that happened a few years back. There were some people who were completely, who disappeared, and we didn't know exactly where they ended up. A lot of people were found here and there without knowing exactly who they are, where they come from. So using DNA profiling, we can actually find out where the people come from, at least looking at the genetic background. Nowadays, We can do food analysis and determine authenticity of the food. For instance, there was a batch of meat in the US that were thought to be cow meat, beef, right? But genetic analysis show that that restaurant that was presenting cow meat as cow meat was not cow meat, but it was rather horse meat, right? You know that in many places in the world, horse is used as meat, right? While in other places in the world, horse is not used as meat primarily, right? So in that particular instance, we saw that the restaurants were cheating, right? Presenting horse meat as cow meat. And finally, but not least, we can actually find a lot about our history, right? DNA profiling in that case actually works like a time machine. Okay, now when we look into the background of President Jefferson, nowadays it is clear that President Jefferson actually had an affair or was sleeping with one of his slaves called Sally Hermings, right? So Sally Hermings had a number of children. When we look at the DNA of the offsprings of these children, now what was at one time a rumor that Thomas Jefferson had children with her slave is not a rumor anymore. There are scientific evidences with genetics evidences that indeed this offspring actually have DNA that can be traced back to Thomas Jefferson. So this day, you have members of the family from that slavery origin and the Jefferson are now holding annually family reunions. So now we know, right, that for sure Thomas Jefferson had many children, not one, not two, many children with some of his slaves. So here we have a picture that shows actually a family reunion of the Jeffersons as they meet yearly. And you can also see the different tones of skin in this family. In precision medicine, we are trying to be as targeted, as specific as possible. We know today that a drug that works for one person may not work with the same efficacy in another person. Well, this is because we are genetically different. And this is what we call pharmacogenetics, right? Pharmacogenetics tells us that some drugs can be more effective in some individuals, while the same drug is less effective in other individuals just because of the difference in the genetic background between these individuals, right? So we look at genomes, we compare them, and we can tell when looking at the genome of one particular individual, one patient, that we should not use, for instance, as pain medication, morphine, in this patient because we know that his genome analysis shows that that person may be sensitive to um, to I'm drawing a blank, to morphine. There we go. Okay, so we are evaluating data, genetic data, to actually come up with very specifically targeted treatments. Today, we look into the microbiome. The microbiome, it's basically all the microorganisms that are found in you and on you. You can have a genome of all that entire microbial population. And we know today that your microbiome plays a very important role in your health, right? Living in certain condition is going to cause you to be prone to some diseases because of your microbiome, right? So knowing specifically the type of microbiome you have can be actually a solution to solving some issues in terms of treatment. Now, because of, again, the knowledge that we have about genetics, it is possible to do a genetic modification. This genetic modification consists of altering DNA, move DNA from one origin to another, for instance, from a human to a bacterium, from a plant to another plant, okay? And by doing so, we produce what we call genetically modified organisms. These genetically modified organisms allow scientists to produce drugs, for instance, antimicrobials, in large amount that we would not be able to produce naturally, right? We are able to produce human insulin using bacteria, right? We are modifying bacteria to produce human insulin, and therefore we can address our diabetes issue in the community. Also, there are some newer technologies, including genome editing, that consist of replacing a defective gene with a normal gene. With genome editing, we can remove a gene, we can add a gene, we can alter a gene, we can replace gene. One of the most common genome editing approach is CRISPR-Cas9, which is very commonly used. Today, because of the technology that we have, we can focus exclusively on the coding part of our genome. And when looking at that coding part of our genome, we try to look at one particular chromosome and we ask the following question: how are exomes aligned on that chromosome? In which sequence these exomes are aligned on the chromosome? This allows us to know exactly what gene is carried by which chromosome and which disease may occur from which chromosome, right? That exome sequencing is becoming more and more important these days, because we can actually determine if the proteins are wild type copies of proteins or mutated copies of proteins, so we can know if an particular individual is producing an alternate form of a particular gene. Okay, exome sequencing is being done, and very importantly, that exome sequencing, while it was taking many years to achieve, can now be done very quickly in a matter of hours. In so much that a patient who comes in the hospital without knowing exactly how to treat that patient, by doing this exome sequencing in a short period of time, it is possible. When I say short period of time, again, we're talking about hours, right? Not years of studies, right? We can actually define ways to treat that patient. Finally, but not least, what is the global perspective when it comes to genomes? Well, the global perspective is going to lead to a topic that we call metagenomics. Metagenomics is when we look at sequencing all the DNA in a given inhabitat. Now, many developed countries and many affluent laboratories are doing this large scale DNA analysis. However, one thing that has been observed is that any finding associated with this metagenomics is now being patented, privatized. Basically, it's my findings. I'm not sharing it with anyone, and if you want to use it, you have to pay me. That is a problem. That's why genetic prospecting now is being limited. So you have ethics committees that are looking closely at what scientists are doing, because with affluent laboratories finding all this information and making it theirs, and saying, well, if someone else wants to do some research on this microorganism that was found in this or this place, you need my permission. That is becoming a problem, hence the limit of genetic prospecting. Genetic is basically a discipline, as you understand now, that affect a large proportion of our life and our environment. So we are all affected. So it's worth, because we are all affected, to wonder, okay, what should we do and what should we not do, right? What is acceptable and what is not acceptable? For instance, we have access to genetic tests and treatment that is not equal across the globe, right? We have the misuse of genetic information. We have abuse of genetics that can cause harm to people. That is a major issue that we need to address. So, genetics and genomics technologies can tremendously improve the quality of our lives and even the quality of our environment, right? The human genome information has been a tremendous source to perform diagnosis, to treat some diseases, to develop some guidelines as to what we should do to strengthen how we approach genetics, specifically human genetics, which finally and again brings us to bioethics, which is what can we do? What can we not do? Who is getting access to the genetic information and who should not get access to the genetic information? Is the government allowed to do whatever they want with our genetic information? What is it? right? So this is going to conclude this chapter. Thank you for your attention, and I will see you to the next chapter.